All right, thank you. So is the volume on this pretty good right now? Can everyone hear me? Is it better now? Okay, and I'll try not to move my head too much too. So I'm really glad to have the opportunity today to share with you some of the work I've been doing in Evan Eichler's lab at the University of Washington. So current human pedigree-based recombination maps do a really great job at detailing the distribution and frequency of crossing over across the genome. However, these high-density recombination maps don't really provide a complete picture of recombination, and this is because they're insufficient to provide resolution to actually detect what we've heard about today many times is actually the most frequent outcome of meiotic double-strand break repair, and these are non-crossovers and gene conversion tracks associated with synthesis strand-dependent annealing. So specifically, these gene conversion events are detected in genetic data as simple sort of copy-paste-like mechanisms. So a small section of one homologous chromosomes gets copied and pasted into an allelic position on the acceptor homolog. And so these gene conversion tracks are very small. They measure less than a kilobase or so, and as Francesca told us this morning, often more on the order of tens of base pairs. So with the resolution of existing pedigree-based maps, we're not able to detect these things. So despite their subtle genetic signature, though, these gene conversion events play a really important role in both evolution and genetics. So gene conversion is a really important mechanism shaping haplotype variation in populations. Specifically, it erodes fine-scale patterns of linkage disequilibrium while keeping broader-scale patterns of linkage disequilibrium intact. What's more, we also know that gene conversion is a major determinant of GC content evolution across the human genome. It can also mimic the signal of positive selection, leading us to detect false positive signals. And it's also an established mechanism of genetic disease. So it seems really important to try to document the distribution of these gene conversion events across the human genome. So one of the objectives of my postdoc has been to try to take advantage of existing whole genome sequence resources and whole genome sequences that are coming offline in humans to try to develop even higher density recombination maps in humans based off of whole genome sequencing. And the idea here is that if we can genotype literally every single heterozygous position in a genome, then we should be able to detect every single recombination breakpoint that can be detected. And this will allow us to define crossovers and define potential gene conversion tracks with maximum possible resolution. And so my initial thought was that if we had some way to derive genome-wide haplotype information for a dad, a mom, and a child, well, then we could simply just march along a haplotype in the child in order to basically just identify places where it switches from copying, say, one paternal haplotype. Sorry, I'm shaking really badly. Where it switches from copying one paternal haplotype to the other. And this would allow us to really straightforwardly and easily identify both putative gene conversion and crossover events. So Jacob Kitzman, who is a graduate student in Jay Shindori's lab at the University of Washington, recently developed a clone-based sequencing method that allows us to resolve haplotype phase genome-wide. And so I'm going to walk you through that method. And for what we've done, we've started off with back clone pool libraries, although it's possible to do this with phosmid pool libraries as well. And so what this library is composed of then is a bunch of back clones that are kind of randomly scattered across the genome. So here I'm showing you a cartoon of one chromosome, and these are the back clones that are tiled across the chromosome. So one thing that's important to know is that any particular back clone contains an insert that's derived from just one haplotype of one of the two possible haplotypes present in the individual. So if we then take this back pool library and then just kind of randomly partition it up into a number of sub-libraries or pools, well, then it's going to become really infrequent and really rare that we would actually uncover within any particular pool back clones that map to the two alternative haplotypes at a single locus. So then if we go ahead and individually sequence these pools, we'll be able to derive haplotype sequence for the regions of the genome that are covered by a clone within that pool. So we start with a single clone library. We divvy it up into a number of pools, and just for reference, we typically take our pools, our libraries, and divide them up into like thousands of pools. So once this has been done, we can barcode our pools uniquely. So all of the clones within this green pool here are going to have an identical barcode. And this barcode is going to be distinct from the barcode that's affixed to all of the clones within the blue pool here. And so once these pools have been barcoded, I can then pool the pools, and we can perform sequencing en masse. And when I get the sequencing back, I'm able to use that barcode sequence to figure out which pool each sequenced read belongs to. So in this way, I can take all of the sequences that map to the green pool here, and I can map those back to the human reference genome. And then I've developed a very simple algorithm that goes along and actually identifies the clone coordinates, the clone boundaries within a given pool. And so once I've identified where the clones actually are within the genome, I then go ahead and just call bases at known heterozygous sites in the individual for which this library has been developed. So we have generated back clone pool libraries for the 3 individuals in this trio here, and this is the NA12878 Trio. Now, back libraries are really, really difficult to construct. I don't understand why, because I've never done it, but there are literally a handful of people on the planet who can develop high-quality back libraries in humans, and I'm very lucky to actually sit right next to one of them in the Eichler lab. So this is a clone pool resource that was developed by another postdoc, Francesca Antonacci. So the NA12878 Trio is part of the Ceph panel, and this is one of the most well-studied trios in human genetics. So the individuals in this trio were sequenced to about 40x by the 1000 Genomes Project. Complete Genomics gathered independently 60x whole genome sequence for these individuals, and Illumina recently released an additional 30x whole genome sequence for this individual here, NA12878. And just for the rest of the talk, for ease, I'm going to call these guys dad, mom, and child rather than those alphanumerical labels for ease. So we've performed a variable amount of sequencing on these clone pools to date. So for the child, we've sequenced a total of 288 pools at this point, totaling over 70,000 clones. The average clone insert size is just shy of 140 kb and was sequenced to a depth of about 6x, although I'll point out that there is tremendous, tremendous variance in this number from clone to clone. Altogether, these pools span about 90% of the human genome, so we have excellent coverage. And for your typical site in the human genome, we have over 4 clones mapping to that particular spot. So we have performed less sequencing at this point for the 2 parents, and additional reads should be coming off the HiSeq any day now. Correspondingly, we have fewer clones in these individuals, reduced genomic coverage, and fewer clones that map to a typical site. So once I have mapped all of these clones and identified their boundaries, the next step is to actually go ahead and call sites at known heterozygous positions within these clones. And so I just want to take a moment to point out that even if we just have a clone that maps to one haplotype at a particular region, we have the ability to actually resolve both haplotypes. And this is probably quite obvious, but I wanted to take a moment to point it out. So we have diploid genotype information for all of these individuals. Recall we have all of these whole genome sequencing resources that are publicly available that we can lean on for this purpose. So here I'm showing you the sequence of the clone vertically here, and here are the diploid genotypes at those sites. Well, so if we have these 2 pieces of information, it's incredibly straightforward to just kind of fill in the blanks and figure out what the haplotype sequence is on the other clone. And I'll point out too that if one of these diploid genotypes is informative for the parent of origin, well, then in this way we can also sort out which clone was, or which clones are carrying a maternally inherited haplotype versus a paternally inherited one. In reality, though, it's not so pretty, and this is because the sequence coverage of so many of our clones is really, really low. It's so low, in fact, that I can't very confidently call bases in these clones. Occasionally, too, I get things like this, where I uncover a heterozygous site in what should be a single haplotype, so that's impossible. These probably represent sequencing or base errors, and probably more rarely, these actually reflect instances where within a given pool, I've got clones that overlap the 2 alternative haplotypes. Now, unfortunately, most of our clones are uninformative in this way, owing again to the fact that we have such low sequencing coverage across most of them at this point. So here on the y-axis is the total number of clones that we've identified in these pools for the child and the 2 parents. So I'm going to ask you to focus first here on the 2 parents. So here, of course, I don't have parental information, so I can't distinguish haplotypes that are maternally versus paternally inherited. But I have the ability at least to distinguish the 2 haplotypes in these cases. Unfortunately, the minority of clones are informative in this way, indicated by the red portions on these bars. In the child, since we do have the parents, I can discriminate between maternal and paternal haplotypes. And again, only about 40% of our haplotypes are informative in this way. So what I do next then is I look at regions of the genome where I have an informative parent clone, and then I ask whether or not there's an informative clone in the child that maps to that same locus that carries the haplotype from that parent. So here I'm showing you a clone from the mom, and although there's a single clone here, I'm able to resolve both haplotypes from that one clone, which is why I've shown it here twice in the two different shades of blue. So here's a clone from the child that maps to the same locus. You can see that it begins by copying the light blue maternal haplotype, and then switches to the dark blue haplotype, indicating that there's probably been a crossover event here in the meiosis of the mom. Oh, and I'll point out too that we obviously don't have information about which maternal haplotype is carried here in the regions where the two clones don't overlap, which I've just denoted in gray. So here is a similar event on chromosome 1 in the father, where we detect putative evidence for a gene conversion event. So here you can see across the region of clone overlap, we begin by copying the dark green haplotype, and there's a little blip of light green haplotype in there before it switches back to the darker green haplotype, indicating a possible gene conversion event during paternal meiosis here. And I'll point out that this blip of light green measures about 1,200 base pairs, which is probably a bit long relative to what Francesca told us this morning, but it's probably not too ridiculous. So at this point, we have informative clones in both the mom and child that allow us to actually survey 19% of the human genome in this way. And we have a similar fraction of the paternal genome that I'm able to examine recombination in as well. Now you'll note that despite the fact that I'm only able to look at a small fraction of the genome, I'm able to detect a lot of recombination events. Now recall that these are, I'm looking at single meiotic transmissions here, and I'm sure as all of you appreciate, these numbers don't really sit very well with what we already know about meiotic recombination in humans. So we know that there are a couple hundred double-strand breaks that form at the onset of meiosis. Fewer than 100 of those are resolved as crossovers, maybe a couple hundred gene conversion events. So all told, across 20% of the genome, I've uncovered orders of magnitude more recombination events than we'd expect across the whole genome. So one thought I had is that, well, we have really low coverage across many of these clones. Maybe genotyping errors and base miscalls are sort of masquerading as false positive signals of recombination. So if I go ahead and just limit myself to instances where we have multiple clones that validate or show the same pattern of haplotype inheritance, maybe that'll help those numbers drop a little bit. So that's what these numbers here are showing. And here's a good example of a crossover event that's validated or consistent with two different clones. And you can see that these numbers do drop to a fraction of their original totals, but they're still way, way too high. So even after filtering out events that are supported by just a single clone haplotype, we still are getting numbers that are really, really noisy. I suspect what much of this represents are cell line artifacts. So these libraries were constructed from cell line-based DNA. And most of our genomic resources in humans, including the HapMap and the 1,000 Genomes project, most of these DNA samples are derived from cell line-based material. So cell lines are effectively cancers, and we know that they have aberrant mutation and recombination profiles. And I suspect that a lot of what I'm detecting here reflects sort of in vitro recombination events that have occurred during cell line maintenance and cell line propagation. So it's unclear to me how useful this particular resource is going to be for telling us anything about meiotic recombination, but we're holding out that it might be useful for saying something about the distribution of mitotic recombination in humans. So in summary, these clone pool libraries can be used to experimentally phase whole genomes. And in theory, this should be a strategy that will allow us to resolve the distribution of gene conversion events and crossover simultaneously in single meioses. To do this, though, I think we're going to need to sequence clones to a greater depth than what we've already done. And I think we're going to need to apply this method to DNA that's not derived from cell line-based material, as what I've been able to find so far and is kind of emerging as a common theme of my postdoc, is that the haplotype structure of cell lines is really atypical. And so as I've said several times now, we are performing some additional clone pool sequencing for the 2 parents, which might help increase clone coverage, allowing us to identify more noise probably in this data set. But we are still kind of holding out for the possibility of using this data to examine distribution of mitotic recombination in humans. And along with Mario Ventura, a visiting faculty member in the Eichler lab, and his graduate student Claudia, we've generated phosmid clone pool libraries for 4 gorillas, shown here in dark blue in this 3-generation pedigree. And our hope is that we might be able to extend this methodology to survey fine-scale haplotype inheritance in a great ape species. So with that, I'd just like to thank the people who actually constructed the libraries I worked with. Francesca, and then Mario and Claudia generated those gorilla libraries that I'm in the process of analyzing right now. And I'd like to acknowledge NRSA training grant to the Genome Sciences Department for funding, and thanks to my boss, Evan Eichler, for support. And thank you.